Good afternoon, everyone. My name is Mark Nardone. I'm director and co-founder of Bio-Trac. Welcome to our Thursday weekly webinar series. We ask that questions be submitted in the Q&A box, and they'll be addressed at the end of the presentation as time commits. For those who might not be familiar with our program, Bio-Trac offers hands-on training workshops for research scientists. These workshops, which are team taught by active researchers, focus on the latest relevant techniques that are necessary for laboratory research. Originally based at the National Institutes of Health for 30 years, Bio-Trac is now located on the Montgomery College Germantown campus in the Bioscience Education Center. Workshops are open to the scientific community. Since the program's inception, we have provided hands-on training to over 17,000 scientists. Along with our hands-on training workshops, we continue to serve the scientific community by offering bio panels, custom designed training programs, weekly webinar series, hosting government and industry retreats and symposiums, as well as other key scientific events and outreach initiatives, such as the Maryland Science Olympiad. Today, it's my pleasure to introduce to you Dr. Richard Lee. Dr. Lee considers himself a Johns Hopkins University lifer. He received his BS in biomedical engineering and his PhD in molecular biology and genetics at Johns Hopkins University. Currently, he is an assistant professor in the Department of Psychiatry, and his main research interest is in the elucidating of epigenetic mechanisms of the HPA axis and the neuroendocrine hormones in the context of psychiatric disorders. As director of Bio-Trac, I've had the pleasure of working with Richard for many years, as he is a key contributor to our Advances in Epigenetics training workshop. This workshop has provided hands-on training to research scientists from local, national, and international institutions. So without further pause, I'd like to introduce to you Dr. Richard Lee, presenting on Bisulfite Pyrosequencing, Quantitative Approaches for Assessing DNA Methylation. Richard, they're all yours.
Thank you, Mark, and thank you so much for this invitation, Bio-Trac webinar. Hello, my name is Richard Lee. I hope everyone is doing well and safe. Today, I will be talking to you about bisulfite pyrosequencing as a quantitative approach for assessing DNA methylation. I worked with Mark for the past several years, and I I conduct a workshop, the last part of which involves a lab where we show the participants how to perform a pyrosequencing assay. Unfortunately, we can't do that through Zoom, but I will show you how to design an assay and kind of show you the beginning steps of bisulfite pyrosequencing by showing you, I guess, the lecture portion of that presentation. So there are, I mean, to be fair, bisulfite pyrosequencing actually is quite an old platform. It's been around for more than 10 years. First started with developing a company called Biotage in Sweden, and then it was acquired by Quiogen several years back. But the pyrosequencing platform still is the gold standard for assessing DNA methylation. And largely because it is a highly quantitative method. So, you know, there are multiple ways of assessing DNA methylation, both candidate and genome-wide. And we'll get into some of this later in the presentation, but it's highly quantitative, and it's ideal, of course. It's the strength and a limitation for candidate CPG or DMR, or differentially methylated region analysis. And it's, I think, in my opinion, more important now because I think that we've moved on from, you know, doing candidate region assays for methylation to genome-wide, and I will show you in the slides later on that oftentimes when you conduct these genome-wide assays, they're not often accurate, and they need some sort of an independent validation for some of the data that gets generated or the DMRs that are predicted from these sequencing endeavors. And I think that pyrosequencing platform is ideal for that purpose. There are some conveniences to studying DNA methylation, but before I even mention the convenience, there should be a rationale in your study or your experiments why you need to look at DNA methylation. Of course, when you look at epigenetic mechanisms, there are at least three main epigenetic mechanisms. We have DNA methylation and its kind of variations, including hydroxymethylation. And then there are histone modifications for which there are, I guess, very many modifications on the different subunits of histones. And lastly, there's the non-coding RNAs. You know, you could look at microRNAs or long non-coding RNAs that also regulate gene function as well. So when you are thinking about your project, there needs to be a strong rationale for looking at methylation in the first place. Having said that, you know, DNA methylation analysis as a epigenetic mechanism is pretty convenient compared to some of the other two mechanisms for the reason that, you know, DNA is fairly stable, unlike RNA or proteins that need to be kept at -80. So you could store your DNA samples at -20 degrees. They can undergo, you know, a good amount of repeated freeze-thaw cycles, and they're fine. And it turns out that, you know, for me, when I looked at lots of samples from clinical studies, turns out that, you know, you could get decent DNA in decent quality, but you can't say the same for RNA or proteins, especially from post-mortem samples. And I think that DNA methylation is pretty good for assessing, you know, how long do these epigenetic marks last. You know, you have multiple samples that you could test at once using, you know, platforms such as a pyrosequencer of, you know, different time points, and then get a feel for how long these epigenetic mechanisms last. And of course, you know, when you study DNA methylation, you only need a very small amount of the material. So, you know, the other common method is the chromatin immunoprecipitation, where you need a good a chunk of the tissue or several thousand cells at least to get a good signal. With measuring DNA methylation, you only need about a few nanograms at the least to get a fairly decent result. And of course, as I will try to convince you that detection methods are fairly easy and straightforward. This is something that you guys probably would all know by now. So DNA methylation, just briefly, involves addition of a methyl group to cytosine. Of course, there are lots of different variations, but for the sake of this presentation, I will just talk about 5-methylcytosine catalyzed by the DNMT or DNA methyltransferase enzyme using S-adenosylmethionine as the methyl donor. And, you know, it's much more nuanced, but you could think of, you know, of course, like a promoter on a tumor suppressor gene. If the region is unmethylated, as represented by these kind of white color lollipops, and it's unmethylated, so then it's open for transcription. So then the tumor suppressor gene is expressed. But in cancer, you know, these represented by black lollipops, they're methylated. So then they interfere with binding of specific transcription factors. So then the transcription is hindered. That's a kind of a basic view of how DNA methylation functionally affects transcription. But of course, lots of different, it's, you know, this is a kind of a generalized concept rather than deep, in-depth, obviously, statement. So as outlined here and will be discussed, the pipeline for bisulfite pyrosequencing involves, of course, your source of DNA. It needs to be extracted and the concentration determined. Let me just say that quality and quantity make a huge difference, especially when it comes to bisulfite conversion. You know, obviously with DNA, more is better, but if, you know, if you could get a smaller amount with a better quality, that's probably better than a lot of DNA with not so great quality. And then it involves bisulfite conversion of that DNA, usually about 500 nanograms to about a microgram, but I've gone as low as 50 nanograms of DNA, and you could get very nice pyrosequencing assays run with that amount. And then what you do with that is you get the bisulfite converted DNA using these kits, and then you have primer sets that you use, to target your regions of interest. It might be a promoter of a gene, or it could be like an intronic regulatory region, which are important for gene regulation. And if you're doing, if you're designing PCR product assays, pyrosequencing assays for the first time, you need to run it on a gel to make sure that you get a nice clear band then that you could use to for pyrosequencing. Just briefly, bisulfite conversion of DNA, and once again, I'm going to kind of generalize for the sake of time. Bisulfite conversion basically leaves methylated Cs in the context of CG intact, while any unmethylated CGs or any other cytosines that are not in the context of CGs are converted. So you could see here at the bottom, here's methylated CG, and there's an unmethylated CG after bisulfite conversion. The CG that's methylated is left intact, whereas the CG that's unmethylated turns into uracil, which, for all purposes in PCR reactions is turned to TG. And then you notice that some of the other Cs, cytosines that are not in the context of a CG are also converted, because the idea is that for the most part, they're not methylated. What is important to know when you perform a pyrosequencing assay, and when you bisulfite convert the DNA, is that after the bisulfite conversion, your double-stranded DNA is not complementary anymore. So you can see here a complementary double-stranded DNA, and then, if you bisulfite convert this particular sequence, you can see here the conversion process, and I left just the CGs as they are, as if they're methylated. And then you could see the cytosines turn to thymines, and then the bottom strand is also converted. And then when you align them back together, you could see that they're no longer complementary. So when you design your PCR assays for bisulfite treated DNA for pyrosequencing, pick one strand and design primers against that one. You could choose the top or the bottom, but I've had people in the past who've designed, one 5' primer for the top strand, and then the 3' primer against sequence at the bottom, and the PCR doesn't work. So this is the kind of the kernel of the presentation, which is I'm trying to I guess share with you how to perform bisulfite pyrosequencing. And so over the past, I think maybe like 10 years or something like that, I've had now designed over 200 pretty good pyrosequencing assays, and from probably like about a dozen organisms. But it doesn't matter what organism your DNA is coming from. DNA is DNA. So then once you have a good quality DNA, you could go ahead and do all these steps. But these are kind of the tricks that I've learned to design PCR primer sets for methylation analysis. Now realize that when you design primers, it's gonna obviously bind to your bisulfite converted DNA to start PCR amplification. And you are trying to design primers that flank the CPGs of interest, and hopefully your primers, unless absolutely necessary, will not contain these CGs. That means that all of the Cs in your primer design would have turned to Ts, thus leading to the loss of complexity after bisulfite conversion. So you have 4 nucleotide sequences, and then now you only have 3 because all of the Cs have been converted away to Ts. And as I mentioned to you before, they're non-complementary, so you work with one strand. And it's hard to avoid, but you want to avoid areas with lots of Ts. As you can see in this little sequence here, when you're designing normal PCR primers, I mean, this would be a perfectly legitimate primer sequence. But then from after it's undergone bisulfite conversion, you can see that a lot of the Cs have been turned into Ts, and then you have a whole stretch of Ts that presumably, or I guess experimentally verified, there's Ts everywhere across the genome where there used to be Cs and Ts. And so if you design primers that include lots of T's, it's gonna cause problems with non-specific priming. And I, of course, and I, of course, tried to include about, maybe 5 or 6 G's as possible, because you've lost the C's, which is a source of 3 hydrogen bonds versus A's and T's that have 2 hydrogen bonds. And so what happens is your annealing temperature is going to be lowered even to 40 degrees Celsius. And what that leads to is lots of non-specific amplification. So what you want to do is increase your annealing temperature as much as possible, and that is possible if you include G's in your primer sequence design to maximize the number of hydrogen bonds that will increase your annealing temperature. And then similarly, I also increased the number of primer nucleotide sequence from usually about 18 to 20 for regular PCR to greater than 24. I've really difficult regions, I've gone as long as 30 nucleotides out, just so I could increase the annealing temperature to get rid of as much non-specific priming as possible. And then the last 5 nucleotides of my primer sequence, I do kind of like a search and find within my region of interest to make sure that those regions don't exist, except where my primer is. Because if there's multiple copies of that 5 base pair or nucleotide region, it's very possible, given the lower annealing temperature, that you could have mispriming as well. And then I use 2 sets of primers for specificity. This is because, for all the reasons that I mentioned with the loss of complexity, that you will inevitably end up amplifying other regions, and a lot of your primer will just kind of get soaked up into amplifying these other regions, and you'll get a very small amount of your target DNA amplified. And when that happens, by doing a second set, nested PCR within the outer set, you could actually increase the amount of DNA from your target of interest. And then similarly, I increase the cycling number to get the amount of increased amount of template. And then finally, I use in my primer design also, usually not always, that they're priming off of a converted T. So it would be A in the opposite strand, and then I would then prime off of using a T to reflect a C that's been converted to T in a kind of effort to maximize amplification of fully converted DNA. And one thing to know with bisulfite conversion is that it's never 100% complete, so that there might be areas with C's that actually may not be actually methylated, but it might be due to poor conversion. So this is a nightmare scenario of a CpG island that in this case is the mouse glucocorticoid receptor promoter. And you can see here it's basically pockmarked with lots of CGs. And what you notice here is also there are stretches of C's here, not in the context of CGs, next to T's in some of the cases that will just turn into stretches of T's once you bisulfite convert the DNA. So this is a kind of a nightmare scenario for a bisulfite pyrosequencing design, but it is still possible to design assays to look at specific CpGs. Some of them are you just can't design primers against it, and you're gonna have to deal with that. But some really important regions, if you use clever tricks, then you could actually design some primers even in this region. So just in a crude kind of display here is even after using the advice that I've given you in the previous slides, oftentimes many of your PCR endeavors will not turn out well. So you see this huge smear here even after 2 cycles, two primer sets, and multiple increased cycling number. And then you have some here that are pretty decent. This is the one that I'm going to talk about later on. This is for the MAOA locus. And you can see here evidence of some primer dimers, which you want to have PCR products without the primer dimers because they will have a biotinylated modification, as I will mention to you in the later slides. And then this is, these are two assays that I think provide a pretty decent amplification. You want to get a nice clear band for you to be able to get reliable methylation results doing pyrosequencing. And so this is the essence of pyrosequencing. Without going into the technical details, what happens is when you have, you have a pyrosequencing primer that will bind to a single-stranded PCR product. So remember, you amplified now your region of interest. It's a double-stranded PCR product. It turns out that one of the primers that you're going to use on the inside is going to be biotinylated. And using streptavidin-coated beads, you're going to use that to pull it down into the bottom of your well, and then use like sodium hydroxide to denature. So you have one single strand of DNA with biotinylated, and that's immobilized to a streptavidin-coated bead that binds to biotin. And then you're going to have assay primers that are going to land a little bit upstream of your CG of interest that you want to assess DNA methylation from. So then you'll do, the machine will, you'll enter the sequence of the extension, and as the primer is extending, when it comes to a CpG dinucleotide, you'll throw in a T, and then through a reaction that involves the release of pyrophosphate, and that's where the term pyrosequencing comes from. As the nucleotide is incorporated, when you throw in a C, the pyrophosphates are released, which are used by enzymes to generate ATP to provide light output using luciferase, so it's a luminometric measure. And then you'll throw in a C for pieces of DNA that are supposed to be unmethylated. So you get a fluorescence from, a luminescence from the CG and the TG. And what the software does is to take the ratio of the CG over CG plus TG to get a percent methylation for that particular CpG for all the DNA that are pieces that are participating in that reaction. And so then you could use either multiple assay primers, or with one assay targeting multiple CpGs, you can actually get methylation status of multiple CpGs in an amplicon. We have in the lab the old, I guess the Q96 pyrosequencer, as well as the much newer one. They pretty much are predicated on the exact same kind of mechanism. Obviously, the implementation, the machine is a lot more sophisticated and automated for the newer model. And this is what happens when you have DNA methylation analysis being done by the pyrosequencer. And I'm using the old colony sequencing and next generation sequencing as a comparison. So when you determine DNA methylation using this old colony-based method, what happens is you do bisulfite conversion of your DNA, you PCR amplify, and then you clone it into a vector, and then you do a transformation into E. coli basically, and then when you spread it out on a petri dish on an LB agar plate, and then you get colonies. Each colony represents a single piece of DNA that's been incorporated into the vector. And then you do Sanger sequencing, from both directions to look at, let's say in this case, 4 CpGs. So one colony and the mini prep from that colony, followed by Sanger sequencing, will have, 4 circles, meaning it's unmethylated, and then white circles that are unmethylated, and then you'll have different patterns of methylation. And then when you get, in this case, 4 colonies, and you tally them across, downward, you'll get a rough estimate of percent methylation. Now, this is kind of semi-quantitative at best, because your percent methylation is determined by these numbers. And if you were to get another colony added that has all methylated CpGs, you could imagine how that affects the overall methylation. So this is the old colony sequencing method, but it's very similar to next generation sequencing. In this case, the number of colonies that you pick is equivalent to your read depth of a particular CpG, because you will have sequencing data that will mapped to a particular CPG, and the amount of read depth with the CGs versus the TGs will be used by your statistician to determine what the percentage is. So these methods are quantitative at best. And when you have, on the other hand, when you have pyrosequencing, you have all of the PCR products that could number in the hundreds of millions. And then for all of those pieces of DNA, you are performing pyrosequencing. So this is the reason why it's a much more, or I would say infinitely more quantitative than these traditional methods, even using next generation sequencing. Let me switch gears now to give you an example of an assay that I've been using in the Bio-Trac lab portion. Let me just briefly tell you about the rationale for looking at DNA methylation in the context of psychiatry and stress hormones or glucocorticoids. When I joined psychiatry, I've learned that high levels of stress hormone cortisol, the umbrella term is glucocorticoid, and glucocorticoid resistance are often comorbid with depression. And glucocorticoids are one of the few molecules that can trigger mania and depression in bipolar disorder. And I've also worked with Cushing's disease. So this is these are patients with pituitary tumors that culminate in high levels of cortisol. And it turns out that these patients, 60–90% of them develop depression and anxiety. And interestingly enough, when you either biochemically or surgically remove the tumors or biochemically treat them with glucocorticoid receptor antagonists, these depressive symptoms are alleviated. So it's a very nice cause and effect relationship between high levels of cortisol and psychiatric symptoms. And from my work and those of others have shown in the past, probably like 10 years or so, stress hormone can directly alter DNA methylation. So this is my rationale for looking at DNA methylation as one of the epigenetic mechanisms involved in psychiatric disorders and exposure to chronic stress. So the case study I'm going to briefly talk about here is a gene called monoamine oxidase A. It's an enzyme that catalyzes the oxidation of monoamines such as dopamine, norepinephrine, and serotonin. If you guys, many of you will know, dopamine is intimately linked to psychosis and drug-seeking behavior, like drug addiction. So in fact, when you take a lot of, I guess, methamphetamine, dopamine is one of these things that are released in copious amounts in your brain. And then, of course, we know that serotonin is implicated in depression. And I've started, using this example, this gene as an example, because it's one of these genes that are fairly well regulated by glucocorticoids. This is worked on in 293 cells, but you can see it in neuroblastoma cell lines and in mouse brain cell lines as well, or even in the mouse brain. That treatment with glucocorticoids, in this case, it's dexamethasone, which is an artificial glucocorticoid mimicking cortisol, leads to a robust expression of MAOA in these cells. And so then the question to ask is, okay, given what I've found about stress hormones leading to epigenetic changes, especially DNA methylation, is there some sort of an epigenetic component that's consistent with gene expression changes? And so then I look at the UCSC genome browser. That's the genomic location of MAOA. Now, I'm using this as an example from my own research, but whatever research that you're interested in, if there's a fairly good rationale for looking at DNA methylation in your animal models or in your clinical samples, then this would be kind of the approach to take, is to look at a specific candidate gene that might be regulated. First look to see if there's a functional difference. And if there is, then, are there any epigenetic mechanisms? Now, I have to admit that, until the next generation sequencing platforms came along, it's pretty difficult to find where methylation might be changing. Even in a gene, it turns out that most of the promoters don't undergo epigenetic changes in terms of DNA methylation, at least in my research. And what I found out is that many of these regions that are undergoing stress hormone-induced changes, epigenetic changes occur in intronic regions. And you can see here, there are regulatory regions in the intronic regions of this gene. And then you could also see there's a annotation for NR3C1. This is the, I guess, the official nomenclature for the glucocorticoid receptor. And so then I looked to see, okay, there's a much stronger, presence here. This was experimentally verified, from the ENCODE database. And so then I designed, pull out that sequence. Unfortunately, there's only 3 CPGs, associated with this glucocorticoid receptor binding region. So, the receptor for the stress hormone acts as a transcription factor as well. And you can see here, there are only 3 CGs, but I'm gonna go ahead and see if any of these are epigenetically changed with prolonged dexamethasone exposure. So I'm designing now a pyrosequencing assay. And for the sake of time, let me just briefly mention, I will design 2 sets of primers, orange in the outside and blue on the inside. And then these are in green, the assay primers that I'm actually gonna use to assay the CPGs. The first one will just assay 1 CG, and then the second one will assay 2 CPGs. During the actual performing the pyrosequencing, you will, as I mentioned to you before, denature. One is biotinylated, and then you're gonna land primers here and here in 2 separate reactions to look at this particular C versus T, methylated versus unmethylated. And then what happens at the end is you have a assay. These are done in multiples, and I did triplicate of each treated and untreated for 2 assays, and you could see the pyrogram that provides you with a % methylation at 2 CPGs for the second assay. And so then voila, you have a expression difference with glucocorticoid treatment, and then matching that is a epigenetic change or DNA methylation change associated with these 3 CPGs. And of course, I've also been involved in looking at genome-wide methylation signatures across the genome to facilitate identification of other areas. And this is crucial for research because it allows you to identify gene sets and pathways. But you get at the end of performing such methylation analysis, with bisulfite converted DNA, is that your statistician will give you kind of a list of areas that are potentially interesting and maybe changing in methylation, and then we'll give you, this is a example of one DMR or differentially methylated region where methylation might be changing between 2 groups of animals treated with vehicle versus steroid cortisol. And then what you do then is you look at these areas in pink, which are representing the differentially methylated regions, design pyroassays, and then you validate by pyrosequencing. Now, this is where I think pyrosequencing is essential, at least for me, because in doing these genome-wide experiments, and I've done quite a few, there are regions that sometimes don't validate even when using the exact same samples, and that's a little bit troubling. But in these instances, it turns out that the analysis and all of the processing of making the libraries and so forth were done correctly. And so this was a genome-wide platform available in this case by Agilent Technologies called a SureSelect MethylSeq kit. But there are lots of companies that make these platforms that you could use. So in summary, pyrosequencing is a relatively easy method, I think, to elucidate DNA methylation mechanisms for gene expression. And I think it remains to be the gold standard platform because it's very quantitative for assessing DNA methylation. And I think now it's important because it can be used to validate, independently validate genome-wide experiments. Thank you.
Thank you, Dr. Lee. That was an excellent presentation. Do thank you for your time. We do have one question that was asked. See, the person says, I did a PCR from bisulfite DNA and I got a very faint band in electrophoresis. Do you have any recommendations to improve my PCR?
So as I mentioned to you before, I don't know what number cycle that you're using. I would probably increase it. So I think the standard, obviously the PCR cycle is 40. I do all mine at like 45, 47. So you get tons more of the amplicon. And if you get a nice band from doing that, meaning that you are able to successfully, increase the amount of DNA in that for that amplicon, that's great. But if you start to see, smears or additional bands, then I would recommend that you redesign the primers. Maybe take the primers a little bit out, or the outside primer set, you could even take it out as far as like 500 base pairs, and it would still work. But the inside, has to be relatively small, like 250 to 300, around there, maybe a little bit more. So yeah, I would redesign the primers if increasing the cycle number doesn't help you.
We have another question that just came in. Can you detect non-C methylation epigenetic marks, i.e. methyl A by pyrosequencing?
Yeah, so the software itself does not allow you to do that at this time. I mean, even the newer model, I've just been using it for CPG methylation. I, you know, I, it's predicated on the fact that the bisulfite conversion changes the unmethylated cytosine to uracil. So if additional methylation in another nucleotide does not lead to a formation of a analog, like a different nucleotide, then the pyrosequencer would not be able to detect that. But if it does, if let's say for some, now that this is, my limitation on biochemistry here, but if methylated A can be converted in some way to another nucleotide, then you could definitely use some of the software modification on the pyrosequencer, treated like a SNP, basically, to detect, the percent methylation differences for that particular modification.
Okay, two more questions and then we'll have to wrap up. Next one, would you anticipate differential methylation of cell types within a tissue, for example, liver or brain, with treatments? If so, would pyrosequencing of whole tissue give potentially misleading results?
Yes, absolutely it does. In humans, like I think, a lot of the human studies are done, using these deconvolution methods, especially blood. They have lots of methods, especially for the the Illumina platform, right? You do have these deconvolution methods, but I've also tested individually, especially in mouse model where they're given lots of stress hormone, and there's definitely changes in the proportion of cell types. And so what I was able to show in a publication, looking at the methylation changes in blood, is that there's a proportion of it that's driven by lots of methylation due to stress hormone treatment. But then when you examine by different cell types, it's a combination of both. There's a change in the population of cells. For instance, in the mouse blood, the T cell population shrinks, and then there's a larger neutrophil population with a very different methylation signature at that particular CPG. So it kind of makes you think there's a change in methylation going on there, but it's actually not. I was able to show that it's a combination of both.
Okay. And last question, do we need to confirm every primer by electrophoresis? We cannot confirm by CyberGreen real-time PCR.
CyberGreen real-time PCR. So CyberGreen is would be a horrendous method in my opinion, because CyberGreen just binds randomly to any random double-stranded DNA. So then when you're using biotinylated primers and you have like a huge primer dimer, or even like a whole smear, they're all going to be picked up by cybergreen. So that's not a good method. qPCR, you might be able to get away with it because, you know, there's a melting curve analysis at the end. And so if you get a nice, you know, peak that's reflective of a nice single PCR band, then it might be sufficient. But, you know, instead of running a qPCR reaction, you know, I would just load a PCR product on a agarose gel, and it's done in like, you know, 20 minutes. I'd rather do that and have a visual confirmation that there's a good PCR product there.
Very good. Well, Dr. Lee, again, thank you very much for your time. I'd like to thank everyone for attending today. Just so you know, next Tuesday, you will receive an email from us containing the presentation, a PDF of today's presentation, as well as a link to the recording of today's webinar. Next week's webinar, next Thursday, will be given by Dr. Kristen Bursak from Momentus. And we'll be discussing modeling the prostate tumor microenvironment using organ-on-a-chip technology. So again, thank you everyone for your attention and time. Be safe and look forward to seeing you next week. Take care.
Thanks everyone. Take care.